Mind the gap. Okay, Lauren, thank you very much for joining us. And please remember, everyone, to put your questions in the chat. You're starting at 4:02, so finish at 4:12.
So my name is Lauren. I'm a fourth year biochem major. My PI is Dr. Jamie Van Norman. And today I'm going to be talking to you guys all about IRK and PXE2 homologs in crops that are closely related to Arabidopsis. So I'd like to just begin with a little bit of intro on root systems and how we like to think about them. So typically, when we see trees and plants, we think of roots as a system that anchors them to the ground or that absorbs nutrients for them, like water and sugars. And then also we can think of roots in the sense of agricultural roots, like with crops such as maize. And they would pretty much have the same function in the ways we would think, like absorption and anchoring. But in the lab, we think of roots very differently, or we observe them very differently. So as opposed to seeing them in the way that we see them, like in maize, we see them in these sorts of systems. They're much smaller, and we actually do a lot of study with the primary roots. So that's what my project focuses on, are the types of proteins that localize in these parts of the roots. So if we go ahead and look at the model organism, it's Arabidopsis. It's commonly known as a mustard weed. And we use Arabidopsis in the lab because it's smaller in size, it can regenerate very quickly, and it also is capable of selfing or reproducing with itself. We look at the roots in this system here. And again, it's a model organism in a lab, but it's not practical in nature. It's not agriculturally significant because people don't eat weeds. So we study these systems, and we use this system in the lab, but we make analogs and parallels to it with agriculturally significant crops such as corn and tomato and legumes. So that's what my project focuses on. And when you look at these model organisms, they have this organization. So there's the lateral roots, which are found here, and they're kind of flat and planar, and they're more out to in the horizontal axis. But what we're really interested in here is the primary root. So with the primary root, you have these three different zones of growth. So there's the differentiation zone that you can observe here, the elongation zone, and the cell division zone. And then at the very end, there's a root cap. And when you look closer at this root cap, you notice that there are a multitude of various cell types. So in this gray here, you have the epidermis, and then there are the cortex cells, and then you have the endodermal cells. And the interesting thing about these various cell types is that they're able to be localized by various different proteins. So proteins within these layers of cells, they can exist in various different directions and all throughout the cell, and they help with cell signaling and the development of cells, and they influence neighboring cells in various ways to help the plant grow and develop. So our lab is interested in where these cell types are actually localizing to. So, or where the proteins in these cell types are localizing to. So they can localize to the outside or to the inside of these various cell layers, or they can even be more upward or downward. And depending on where they localize, they'll have a differing influence on how the plant develops. So again, just to restate, our lab focuses on how the protein localization, where it's going in these cell types, how does it affect root tissue patterning? And so one of the major classes of proteins that we're interested in is the localization of transmembrane receptor kinases. So kinases, they function in phosphorylating various substrates, and transmembrane receptor kinases, as indicated by transmembrane, they rest on the plasma membrane of various cell types. And so they'll receive information from other cells, and they can phosphorylate substrates to communicate further into the cell. So in Arabidopsis, there are about 220 of these kinases that exist. And the two that we study are the ones that I mentioned, called IRK and PXC2. So with these two receptor kinases, they're pretty similar. They have a similar function, and they're about 79% similar, and their structure is pretty similar, which is, it kind of parallels with the fact that they have a redundant function. So moving on for a little bit, when we observe IRK and PXC2 and where they accumulate in Arabidopsis roots, this is a confocal microscopy image, and these cell layers that I mentioned before, so there's the endodermis, the cortex, and the epidermis. And with IRK and PXC2, they localize pretty intensely, as indicated by this GFP here. They localize into the outer polar domains of the endodermis, and they're phosphorylating substrate, and they're helping with various communication initiatives between different cell types. And so when we observe that this localization occurs, this is what we expect to see in a wild type image for this type of, for this gene here. And to kind of go on about the function of IRK, we notice that when you mutate the IRK gene and you insert a construct with this mutant gene, there are excessive cell divisions that are in this endodermis layer here. So with this mutant, you're getting way too many endodermis cells. And so this kind of, this suggests that IRK has a function in repressing excessive cell divisions. So just to reiterate, the localization in Arabidopsis of these genes is you have them localizing to the outer polar domain, and that's where they're going. And their function seems to be the repression of excessive cell divisions. And this is for Arabidopsis. And my project focuses on the homologs, which are closely related genes in other plants. So closely related versions of IRK and PXC2 that exist in tomato and maize, legumes, and rice. And so those are the questions we want to answer is, do these homologs have similar localization? Are they going to the same places? And do they have a similar function? Do they repress excessive cell divisions? And so an approach that we have is we have a localization approach. And one of the ways that we want to attack this problem is, or this question is, we're going to create these homolog GFP fusions and insert them into wild type and see where they're going, see where these proteins are localizing. And then our functional approach is the construction of these homolog GFP fusions and insertion of them into the mutant plants to see if the mutant phenotype is rescued. So moving on, so to begin with this construction of these expression vectors, we need to amplify the gene of interest out of these. We need to clone the gene of interest out of the homolog. So you have to amplify these genes via PCR, and then we can confirm the fragment, and we will actually create a smaller vector that holds the gene of interest, and to create a much larger expression vector that actually helps us to answer these questions, we have to perform an LR reaction. So there, you begin with like 3 fragments, one of which being a promoter, which drives the expression vector to a specific portion of the root. And then you have your gene of interest, which is what we're interested in looking at. And then you have GFP, which serves as an indicator. It undergoes an enzymatic reaction, and these portions here, they interact so that all of these fragments, they meet together onto a larger destination vector. And this is the overall expression clone that we're interested in looking at. Then we insert that clone into Arabidopsis, and we're going to insert it into both the wild type, which is Colombia, which will help us answer questions about localization. And then we also insert it into the mutant phenotype, which is, the mutant, which will help us to answer whether or not the expression vector can rescue that phenotype, which answers questions about function. And so we have some prelim data here, and this is what we would expect our expression vectors to look like. And we're noticing that there's localization at the cell division plate. The only difference is in this prelim data is some of the expression is in the vesicles that are more at the center of the cell. We wouldn't expect that, but this might be due to the stability and the fact that the DNA is foreign to Arabidopsis. Overall, the data is very promising, and it's pretty much very similar to what we would expect to see. And that's all I have for you guys today. So thank you for listening. I hope you enjoyed. I'd like to thank my PI and everyone in the lab for all their help. And do you guys have any questions?
That was a fantastic presentation, Lauren. I really liked your slides. They looked great. Did you draw those diagrams of the route?
Oh, no, I didn't draw them myself.
Pretty good. Very pretty anyway.
Thank you.
Do we have any questions for Lauren?
I have a question. So I really enjoyed your talk, Lauren, and I agree your slides are beautiful. I was wondering about trying to, is your lab involved in maybe expressing GFP tag versions of these proteins in the native crop plant, like in tomato or some other species that maybe your lab, has worked with, to try to look in the native system and in case there's accessory proteins that take the GFP tag protein to the correct locale in the cell or in the root?
Yeah, our lab, we only do Arabidopsis, and we're like a root lab. So I'm the only person who works on any other plants outside of Arabidopsis. So I don't think anyone's ever tried to insert into like the endogenous plant, the home plant, but I'm sure that if you were to, there would probably be, like you said, accessory proteins that would affect that localization, because they are they're homologs. So there's you never really know. I don't really know.
Okay, thank you.
Thank you.
So Lauren, when you mention about cell division, unlike Dr. Borkovich, my plant knowledge is limited. However, although they're doing quite well outside, thank you very much. But when you talk about cell division and excessive cell division, cancer comes to mind. And I'm wondering if there's any mammalian homologs for IRK, and also perhaps understanding how it's turned on and off might be useful. Do you find IRK expression elsewhere other than the root tip, or is it turned off?
IRK, I think it's a very highly conserved gene in plants. It has it's a very evolutionarily conserved gene. So I wouldn't be surprised if there were I guess homologs in mammals for IRK, if I understand your question correctly.
Yeah.
Back to cancer and whatnot. So since it is,
we have similarities across immunology, for example.
Yeah.
We get a lot of our ideas from you plant folk.
Yeah. So since IRK is so highly conserved, I wouldn't be surprised if there were genes that were also very similar, structurally to it in mammals, that have a role in these excessive cell divisions, and whether or not mutants of those mutations in those genes or having those genes turned on and off would affect the development of cancer.
So is it suppressed elsewhere in the roots?
In the roots, yeah. There's various regions where IRK will be turned up or down, right. And that will affect whether or not it's how those cells are dividing.
It's very interesting. Any other questions for Lauren from our guests? If not, thank you very much for an excellent presentation. I really enjoyed it.
Thank you.
I don't believe we've had Anthony